ALLERHEILIGEN. About what is in the program. It said talk about model-informed drug development and regulatory decisions today and tomorrow. Sandy?
Hi, and thanks for the opportunity. As we thought about this, when I was first thinking about the talk and our interactions with the regulatory sciences, sometimes it's sort of like a stoplight, is it a go or not? And what we're really wanting to propose is it's much broader. It's many interactions, and I think we've seen that today. So for us, it's still all about the question. And at a very recent Brookings meeting, there were some quotes that I thought were really memorable. Dr. Temple still believes we get the dose wrong the vast majority of the time. Dr. Peck, we still don't understand adherence. We have no ability to translate the clinical trial data to the real-world patient. We're still struggling with this. And I think the part that stabs me in the heart, I heard those two men make that same quote about 25 years ago, and we're still struggling. So I do believe there's a challenge, and I think we've heard a lot of examples today that I do believe there's an answer towards using quantitative approaches. Let me just pause a minute when I talk about modeling and simulation, what that means to me. So when I first got to Merck, one of the comments there was, well, if you need data, what do we need a model for? And so it's really about, these are data-driven. One of the greatest challenges we have is understanding the assumptions that are inherent in clinical drug development. So otherwise we wouldn't have a 93% failure rate. So it helps us clearly define what are the assumptions we're making. And I would argue that's an important step all the way through all of the kinds of models we've seen. They are data-driven. There are a wide variety of data. There are a host of quantitative tools that can be used. And it becomes a portable form of knowledge that we can then use parameter sensitivity analysis to test the most important question: what if we're wrong? And then to go on to enhanced understanding that stems the whole range from internal decisions all the way to patients. So when I talk about integrating models and trying to understand, it's not that models are the center of the universe. They may be my universe, but they're not the center for our patients. But it's a wide variety of models that we need to integrate, whether it's the classic PK/PD exposure response models, more importantly, understanding standard of care. What do we know already about the disease? Can we use much more mechanistic models of disease and disease progression to help us understand how to treat these patients, understanding variability, what makes one patient respond differently, and most importantly in the end, the benefit risk. So I'm going to give you two very fast snapshots. One is we were asked, as Merck has Icentris, and we were developing a chewable tablet. The chewable tablet for these young children gave a slightly different curve in response, and it's not surprising. It's a very different dosage form. But the biggest concern for these HIV patients is you don't want them to develop resistance. So concentrations that occur late that appear to be below that protective range really drive a huge concern. And so the EMA was asking us to develop therapeutic drug monitoring. We needed to establish central labs. And we said, well, can we come and tell you about a mechanistic model that we have built to understand viral load, to understand latent cells, infected cells, active cells, and how those all relate, both for Icentris, but more importantly, all of the compounds on the market understanding HIV. And with that, we were able to build a model that showed if these children receive this dosage form, because we were able to pool data across many trials, where we were able to show if you're down at this range, you are going to see disease progression, or excuse me, resistance developing. What does that mean? And so what we were able to do was to come back and say, look, this, although yes, there does appear to be a slightly lower concentration at the trough level, you can see that this will still be within the range to keep these children safe and not develop resistance. And with that, it overturned a decision, and these chewable tablets are now available to these much younger children. Another example, and I'm just throwing this one in really quickly to highlight that this is for biologics as well, is that we were using these model-informed approaches for Keytruda, which is our anti-PD-1 immunotherapy. And when we were first designing the phase 1, we used our non-clinical data to try to understand the tumor size and what would be the likely needed dose and what would be the schedule that we needed to think about with 2 weeks or 3 weeks. And was so often there was a tendency to want to go to the maximum tolerated dose, which in this case would projected to be 10 mg per kg. So our phase 1 trial, which ended up being 1,000 patients, so it was one of those that just kept adding cohorts as we saw response. What we were able to do then was to use, because we were taking blood samples as well as measuring progression-free survival, overall survival, we were also measuring tumor volume. We were able to, and biomarkers, we were able to put that all together to answer the question, what's the right dose? And it was a 2 mg per kg dose, not a 10 mg per kg dose, which had a tremendous outcome for the compound. So that's today. Where do we, and the organizers challenge us to where are we going tomorrow? And so I've heard today it seems like we've been approaching it from 2 different ends. We have the real world data, the big data. Who would have thought what you could do with Twitter? As we come and think about that data, and then my team that's thinking about it from early discovery to that first in man, to that next step, and on up through regulatory and patient access. So are there new tools that we can use? And I don't have the answer, but we've spent years talking about translating non-clinical to clinical. Is the new world translating the clinical trial so we're predictive from the beginning to real world? And there are some new technologies to help us do this. Can we actually understand the exposure, in the patients at the bedside or at their home? And there's with these new emerging, and some of them are back to the future. Dried blood spots have been around for a gazillion years, and they're back. And we have a way now to let the patient take a card home, a little easy stick. They put the card in an envelope. It has a date timestamp on it automatically, and the data are coming back. What's that mean with all of these new apps? Can we change the trial system to from a site model all the way to the patient? And what would that mean? And one of the things we talked about, and I hope we get a chance to talk about more, is one of the best ways to make these real is real-time simulation and visualization. And that's taking and exploring these models and taking it all the way to the bedside. So changing environment, what are the new intersections? Model qualification, how do we know we've got it right? Would you let your mom take this drug based on the model you just built and the label we just wrote? Technologies to understand adherence. The greatest quote was 50% of the, if you think about the most important covariate is did they take their drug? And how do we pull that out of real world data? And how do we use real world data to help us truly understand disease models and disease progression? Can we actually use data to help us understand what's the right drug for the right patient at the right dose? What are other trial advances? And I look forward to the panel discussion. And thanks.